के नाम से you are online now. Yes. Yes. हाँ जी सर. Welcome, sir.
Thank you.
I'm audible to you now, sir?
भैया दिख रहा है आपको?
Yes. Your screen is visible now, sir. Yes. So, do we start now, sir?
Now we can start.
Yes. Good afternoon, everyone. Welcome to today's session in the webinar series on biotechnology, hosted by the Department of Bio and Nanotechnology of Guru Jambheswar University of Science and Technology, Hisar. It's a great pleasure for me to introduce our speaker today, Professor K.C. Bansal. He has been former director of the National Bureau of Plant Genetic Resource and ICR body in New Delhi. He is a Haryana Vigyan Ratna Award winning personality. Professor Bansal is going to talk to us about recent advances in plant biotechnology, opportunities and future prospects. This is a topic in which we should all deeply interested because it gives us knowledge and wisdom about future directions in plant biotechnology. He is specialized, having spent almost his entire career in plant biotechnology research. So without any further ado, let's move to our today's webinar. A formal reminder for the audience, if you have any question during the presentation, please type them into the chat box. I'll bring them up during the presentation and we'll also have time for question at the end. Now, we would love to hear from Professor Bansal. Welcome, sir. Over to you, sir.
Can you remove this? Can you remove these two people have entered and all this logo keep coming here?
Yes.
I can slide. Yeah. Can I start?
Yes, sir.
Am I audible? Am I audible to all?
Yes, you are audible, sir.
Okay, friends, good afternoon to all of you. At the outset, I would like to thank the organizers from Guru Jambheswar University of Science and Technology and to the Department of Biotechnology for sponsoring this program, particularly for the students of plant biotechnology. And I have a great pleasure in delivering this lecture entitled Recent Advances in Plant Biotechnology and focusing on such and future prospects. And I'm delighted that very many students will be listening to me in addition to several faculty members. And I was told that also Dr. Ashok Chauhan, the president of the MED group of institutions, is also among the audience today. I'm privileged to welcome him as well, as he's the one who is changing the whole scenario of private education in this country and doing work for the country, even outside India at international level. So I welcome you all on board in this webinar, and I will start with this disturbing me, this see waiting room and admit. Mr. Rakesh, please remove it.
Yes, I have removed it, sir.
Can you please remove this?
Yes, I have removed it, sir.
Right, it came again. So, with a historical perspective of plant genetic engineering It's not going to the next slide, buddy.
You click on slide, sir.
It's not going to the next slide.
You double click onto the slide, sir. It will work.
Huh?
You double click on the slide, please.
So I start with the of wherein the first ever methodology for engineering the plants was discovered or was developed by a group of scientists, three group of scientists, with expression of bacterial genes in plant cells, which was actually, technology developed in 1983, and when this gene in plant cells was demonstrated by three group of scientists, Meridel Chilton, Mark von Montagu, and in this slide shown Mark Fraley of Monsanto. First time ever expressed in plant cells. And these three scientists later, 30 years later in 2013, were given the World Food Prize. Actually speaking of engineering plants for agronomic improvement with regard to resistance to various pests, disease resistance, improving the quality. As scientists, improve the crop plants with various traits. So when you can express these bacterial genes, that means not only that you transfer the genes from one, like for example, wheat to wheat or rice, bacteria, or from any other biological organism, still transfer to if it conveys a particular trait of importance. And herein, the expression of bacterial genes was only to select the plants as transformed. Globally, many of the genes from Bacillus thuringiensis, which is Bt genes, were introduced into several of these crops, and also plants were made tolerant to herbicides. So these two different traits, insect resistance and tolerance to herbicides, and later so many traits were. We'll go into the details when we talk of classical plant engineering. So using these tools of classical plant engineering, various traits of agronomic importance were introduced of crops internationally, and the area under cultivation of these GM crops increased from nowhere in 1996 when it just started for to about 190 million hectares. Actually, this has increased more than 100-fold in the last about 20 years or so. The area by GM crops. And here 70 countries as shown here in this global map, 70 countries are growing or taking the benefit of GM crops. Not 70 countries are growing, but 70 countries is the total number. It's roughly about 29 to 30 countries which are developing GM crops and growing GM crops. 40 countries are consuming GM crops or using as food or feed or fiber by importing them to their countries. And you can see here the color in the countries which are importing GM crops, and the green as shown here, Canada or North American continent, or here, Latin American continent, or part of, or even, part of Europe, India, China or Australia, and some of these continental countries, they all in the dark green color are developing and consuming GM crops on their own. So therefore, total about 70 countries are taking the benefit of the GM crops, and which are grown today, and it's increasing every year. The area of 190 million hectares. The crops which have been improved are soybean, maize, canola, which is like our mustard oil, Indian oil, canola grown in European or in Canada, and cotton, potato, eggplant, apple, papaya, sugar beet, alfalfa, squash. See here, for example, soybean, of course, is also used as feed, and canola is used as an oil, which is also processed. Cotton is fiber, but cotton seed oil is consumed. Potato is, of course, a feed. Eggplant, apple is eaten raw, papaya is eaten raw, and like that. So this is a variety of crops which are genetically engineered, which is growing globally, as mentioned, including India, and are being consumed by humans or or food or as food for the livestock. That was the global status. But in India, genetically crops, the one which is commercialized is only one, that is BT cotton, which was approved in two different phases. One was in 2002, that was in mostly Maharashtra region, zone. And then 2005, it was released in northern zone like Punjab, Haryana, and other parts of the country. And together, today, it's grown in about, that is almost the entire area in India where cotton is currently grown. Actually, it's 11.6. And more than 95% of farmers in India have adopted, as you can say, if we see the inception was in 2002. And they're taking great benefit. The production increase in India, it was about 50 million bales, in 2002, and today it is over 35 million bales. It's more than 2.5 times or even 3 times because as a result of introduction of BT cotton. And BT cotton is the gene which is taken from bacteria, Bacillus thuringiensis. It has been introduced and it is now available in n number of different zones of the country, and farmers accordingly are choosing the best hybrid with BT gene into it and taking the benefit and increasing the production. Not only increasing the production, also which is very important. So they ultimately they say, well, I'll show you some of the statistics and how much money is getting saved. After BT cotton in 2002 and 2005, BT eggplant was approved by the apex body in 2010, but unfortunately, there is a moratorium cultivated. Instead, Bangladesh, the same BT eggplant developed by Indian scientists or seed companies, is grown in Bangladesh, and they have entered in, and 4 different varieties have been developed, and Bangladesh farmers are growing and taking the benefit of that particular eggplant. And next crop in India, we are waiting for approval. It has been developed by Professor Deepak Patel, which has demonstrated about 25 to 30% increased production, or productivity time. And this is a kind of a hybrid, which is based on Barnase Barstar kind of, a technology, which is available already in canola, and almost 100 of the canola, which we are talking about, which is GM canola, is based in this technology, and that process being consumed, the oil is being processed, in Canada, where also by the countries which are importing into their countries. So this is based on that technology, wherein GM canola, hybrid canola is based on what Deepak Patel has demonstrated amply, and it has undergone all those biosafety trials, whatever required for the human beings or for the environment, has been recommended, by the Indian regulatory agency. All have been conducted, time and again, and some excellent results. But the only thing is now we are waiting for its approval. Pipeline in India, and after this trait of insect resistance or herbicide tolerance at the international level, or hybrid production in mustard, we would like to utilize the benefits of this technology for improving crop plants. For example, what is happening today in this era of climate change? Our we need to produce more with less input of natural resources like water, fertilizers, or like nutrient is also not expanding anymore. So we need to therefore take the benefit of this technology and introduce traits like nitrogen use efficiency, or in general nutrient use efficiency, and others, or even micronutrient efficiency need to be increased. Drought tolerance or even herbicide tolerance is required, or even salt tolerance. All these abiotic tolerance traits are also important. While they are complex traits, not very easy to genetically engineer through these technologies, but nevertheless, in India, even several crops for resistance to abiotic stresses in a network mode, which was funded by the ICAR, that is National Transgenic Network in Crops, of which I was we, in fact, cloned several genes for various abiotic stresses as shown here with drought, high temperature, etc. And those genes actually were cloned from land races or crop oil relatives in case of wheat or in case of brassica species. And then through genetic engineering approach, we transferred them to various crops like, about again in a network mode by various institutions like rice, wheat, mustard, tomato, groundnut, etc., or even chickpea, pigeon pea, etc. Tolerance were transferred, but those are still, to be taken forward with regard to regulatory agency requirement in India. So apart from what I'm saying is that research is still going on, or several of the GM crops are in pipeline, to go through these regulatory, biosafety trials and to be finally commercially released. Permit us to do that. And I think we would urge the government of India for the benefit of farmers and for the benefit of our own nation. It is important because it gives, as you can see here globally, the market value which has given a benefit in terms of US dollars. You can see USA has got maximum benefit. 2013, for the year 2013, USA is highest because that's the area, out of 190, 40% of the area is in USA, and followed by Argentina and India, where it's about 11.6 to 12 million hectares with single crop BT cotton is also benefited of about over $2 billion US dollars if you see the data of 2013. Similarly, all other countries. In India, I said before, only BT cotton is commercialized, and this cotton, you can see here the cotton, produced out of BT cotton. And this is the produce you can see here out of non-BT cotton, which is damaged. And as a result, what happened, even the cotton seed is damaged, because of the insect is released, that the oil is really, its market value is decreased tremendously. These are the two pictures. I took it from a farmer who is directly growing this BT cotton in this field, and his message is that for BT cotton in India, which is so good and which is so clean. And globally, if you see the benefits, the crop yield increased by 20%. Talking about it, the data over 20 years of analysis, increased crop yield by 22%, increased farmers' profit by profit, and as a result, national economy benefits to the tune of 186 billion US dollars. This is from 1996, 20 years later. And there's important fact is, again, this is so important, conservation of land and biodiversity. Because if you want to double or quadruple the production of anything, you are not able to increase productivity because of these efforts of science and technology. The only option you have is to increase the land under cultivation. But if you don't want to increase the land, or you don't want to cut the forest, or you don't want to, increase or sort of land use plan, if you have land, only for rest of the purposes, and we don't expand the land for agricultural practices or agricultural crops, I think we need to have such technologies. Some of these technologies I'm talking to you about, that is genetic engineering. So conservation of land and biodiversity, both are very important, and which has been observed in these last 20 years. These GM crops almost 100% in some of these areas. And also there's a reduced use of chemical pesticide by 37%. I was talking to you that because of the increase in yield. In India, there's an experience of BT cotton alone, where there's an increase in yield, and also similarly, there's a reduced chemical pesticide use, like compare all the crops. And because of the reduced chemical use by the farmers, naturally the consumers least exposed, and there's a benefit to the environment as well, because those friendly insects will not die, which was a great use of insecticide. And therefore, there's an increase in biodiversity. If I give you an example of honey bee in India, the population after the introduction of BT cotton in some of the states increased tremendously, because otherwise it was getting killed through the use of pesticides. But now, because of the introduction of BT cotton, the pesticide use has reduced, the population of honeybee increased, and the biodiversity also increased. I mean, more and more, species were conserved, and this gives you a total benefit with no negative impact reported so far in 20 years in any of these countries growing or consuming, as food or feed or for whatever reason in various. So now, actually, this was only to give you a summary that what benefits we have harvested through the conventional, I use routes. Those were that you take gene from any source, from a bacteria, from any biological organism I mentioned before, and you can introduce to plants. I'm not describing the method because, I'm talking of a technology which was discovered in 1983, which is quite mature, and I'm sure even the students know, talk to them in their classrooms. But if they have still any questions specific regarding how the genes are transferred, etc., various methods are there, I can talk to them, my references. But then, since the topic deals with the recent advances in plant biotechnology, I thought I would like to familiarize the students and the audience with what are those. Currently the method we are talking about it, which has the ability to do the gene editing coding in the plant itself. I mean, there is an ability now available with the scientists that they want to mutate a particular gene of a particular crop. Plant gene engineering, we were talking about it, transferring gene from bacteria, let us say, to the plant. But here we are trying now to mutate plant's own genes through the tools of genome editing. And just to give you a little background, several years ago, before even conventional breeding, we were trying to induce generation into some of these crop plants by use of various chemicals, mutagens, or through radiation. And that will mutate the genes, and then there will be a selection of populations, and it will take about 10 to 15 to 20 years before those mutant population will stabilize for a particular trait. Then we do the selection, and there are certainly, of course, an advantage of those. Hundreds of varieties in various countries globally were released as a result of those mutation breeding. But that mutation breeding was time consuming, blind the street blindly, and was not precise enough. And any mutation will occur at random. We will not know where it's happening. But scientists are endowed with this kind of a technology, which is genome editing, wherein precisely a single gene or multiple genes in a can be mutated. So, and this technology is quite precise and targeted. Earlier, I mentioned to you about the mutation breeding that was but targeted to a particular gene of interest, but similar to natural mutations, that you will not be able to know any difference between the targeted its wild counterpart. And regulation is also, several of these countries have classified genome edited as non-genetically modified. I give you an example, for example, later, and which is entirely science-based. But then, and speed is substantially faster. Within a year or 2 or 3, you can have the crop variety which is mutated a particular trait, and it is ready to be used even as a donor or the source for transfer that particular trait, through that mutation to any number of high-yielding genotypes transferred to already high-yielding but susceptible genotypes of a particular crop. Cost of production will be less compared to GM crops, because here it is, I said before, country said if it is a non-GMO, no regulation at all, so that will be having significant reduced cost of regulation. And I said before, now number of crops are donated, like for example, rice, wheat, soybean, maize, potato, tomato, and several other crops. And recently, I'm very happy that India is also taking the lead in genetically editing these number of crops. I'll give you one or two examples of that as well as I go further. Well, this technology, in fact, is as scientists make these discoveries by accident. And this is also, I believe, of that one of that kind, wherein, you know, several scientists were puzzled because it's called as CRISPR in a bacterial system, and bacteria has this system developed over time to protect itself from, you know, from virus as invaders. It is now possible to precisely create a double-stranded break in a DNA, okay, and then this double-stranded break is, there is a plant which is utilized as non-homologous end joining. If there is a break, these two ends are joined, and this finally creates some variable length insertions. Variable length insertions or deletions are created. If this particular gene is functional, it becomes non-functional. As a result, this is what is basically a mutation. This is here of a given gene, and that gene, if it was functional, it becomes now non-functional, and there are several traits which are controlled by, you know, these loss of function. There's a positive trait, you know, I give you a few examples. So here, this is the kind of a technology which USDA regulation says it's a non-GMO, because you're not putting any transgene from anywhere. You are only mutating few bases of a gene or adding, and it's a natural phenomena occurring in a plant cell or any. The cells has a capability of repairing this double-stranded break, and as a result, there is an insertion or a deletion, there is a mutation, and there's a loss of function. And this is with several advantages. I'll give you one or two examples. That is one way. Now, second is, as a result of, you know, this guide RNA, single RNA, there is a CRISPR RNA which is designed, which is targeted to a particular sequence of the gene which we know, okay, and you can few bases of that gene coding sequence or in the promoter sequence. You can precisely mutate those genes by having a cRNA targeted to those. The guide RNA will channelize the movement of this Cas9 protein, and it will cause this double-stranded break here again. And then this is how a repaired in their system, because here you are introducing the genes, the bases you want to add or mutate, you're doing that. And also it is possible that you can insert in case of plant genetic engineering and develop transgenics. That is also possible through homology dependent repairing system, that you can introduce a whole new gene altogether. So there are different 1, 2, 3. One is of this kind, which is non-GMO kind by many countries, including US. And two is where you mutate only, let's say 4 to 5 to 10 to 20, you know, whether in a coding sequence of the plant genome itself or the promoter sequence of a given gene. And third, a new gene altogether, that becomes, of course, a transgenic, and we still call it, it will undergo the regulation as we have got in any country for that matter, you know, of regulation. And these are again precise insertion. Here, compared to plant genetic technology, which I talked to you before, the advantage here is that you are inserting a new gene. If you're doing so, a location onto a genome or onto a particular chromosome, because that was not possible. That was random insertion when we talk of plant genetic engineering. Here, therefore, it is, you know, and therefore it's so important. And these are the two, you know, lady scientists who really deserve kind of honor, you know, who showed this kind of bacteria, you know, could be utilized for crop improvement or improvement of, you know, for example, all other organisms where we are interested in. So I just give you ABA receptor is something very important. You know, earlier we knew about the role of ABA in conserving moisture by closure of, and many other, roles it has, particularly in making plants tolerant to drought or range of abiotic stresses. Like, give kind of a tool to the plants whether to allow losing moisture to the air or allow plants to arrest its growth if there is any kind of a stress, or so then the ABA levels will increase, and there are ABA receptors which are act in the way it has been physiologically acting. So very recently, I think it was 2009, that first time someone discovered, which is able to bind to this, ABA, and they've given a whole mechanism by which ABA exerts its mode of action onto the plant physics, moisture or seed dormancy, etc., etc. So the scientists who discovered, actually two groups did that. One of the groups of Jiakang Zhu, they found that there are 13 ABA receptors in rice. And ABA receptors, all 13 probably could be involved in different mechanisms of action for the ABA to act at the whole plant level. So here they mutated. For example, these are the different ABA receptors, 1, 2, 3, 4, 5, 6. They mutated. Editing is also possible now. There are various, Cas associated or CRISPR associated proteins like CPF1, which allows you multiplex targeting. So they're out of 13, they mutated 6, and they found that compared to the wild type, these plants are growing better. And then, because possible permutation combinations for genome editing of these 13 ABA receptors, which is a very big task, actually speaking, But then they selected few, and I that when they mutate 6, there is an improved growth, as you can very clearly see here. But then out of these 6, they found that 1, 4, and 6 even are better, giving a better, still better compared to either wild type or where all 6 were mutated out of 13. So this is because they are able to have a balance, of higher growth, moisture, and this is true. Stomatal opening, stomatal conductance, photosynthesis, water loss. As physiologists, they have all demonstrated it, that if there's a balance between the goal in allowing the plant to arrest its growth or allow it to grow further, So therefore, if you strike that kind of a balance, you see here there is a better growth in these generally. And here yield per plot, as they have shown here, in wild type it was 1500 grams. Here it's about 2,000 grams. It's about 30 to 40% increase. I can say, of course, it's a small plot, but even if you demonstrate it at field level on a large scale trials, it will still amount to be at least 15 to 20%, or even 10% advantage, which today what we are observing is no more than 2 to 3 to 4 to 5%. That means. So this is just one example. Actually, in my own lab, we were fortunate. We were the first ones, again, I would say, to clone an ABA receptor gene from a landrace of rice, which is highly dominant. We could demonstrate through functional validation of this gene in a model plant species Receptors, that this gene, when it is overexpressed, provides advantage to cold stress than the wild type. And they have got longer root system and higher biomass, So, and this was also in 2009. We did not know that time, really speaking, that whether it's ABA receptor. Only when it was discovered in 2009, and even several years later, we realized that the gene which we had, deposited in NCBI happens to be the first of its race, highly drought tolerant, and which was then later demonstrated by Dr. Vishwanathan. Actually, Dr. Vishwanathan from India, head of the division of plant physiology, IRI, happens to be in discovered globally, first time, published a paper in Nature, what kind of those ABA receptors are. So he, in fact, then overexpressed the gene which we cloned from Nagina 22 in rice, Indian mega rice varieties, and found that this gene, which was later named as PIL10, when we cloned it was named as PIL3, provides tolerance but also drought tolerance. And here, some of these plants recovered. These are overexpressing this particular ABA receptor, and here is the wild type. Wild type, they don't recover at all after a number of these different, overexpressed lines. They all survive very well. And this is in rice, in one of the mega varieties of indica rice. And another gene, genome editing, actually. That was what I showed you here was not genome editing. This was through genetic engineering. But I just give an example of one of our own genes, which has been shown to be ABA receptor. Through genome editing of rice, again of a variety of rice, a mega variety, which is listed here. I can't see it in my presentation. I hope you can. And here in one transcription factor encoding gene, loss of function gives, here larger flag leaf size. You can see here flag leaf, and this is the genome edited line with that particular gene. And here you can see the flag leaf width has significantly increased, flag leaf area has significantly increased, and so ability or the recovery of the plants, this is the wild type, and this is the recovered plants are better, and survival rate and against the salt stress 20%, here is more than 60% in the genome edited plants. This is also to do some physiological mechanisms. I don't have time to go into the details of those mechanisms, but and kind of glimpse of some of these recent advances through the use of the genome editing technology. To continue with genome editing, one of the best examples cross culture is actually, there is a bacterial leaf blight disease, which is quite serious, which causes the great damage, to the rice plants. And of course, known for it. And, an Indian scientist, Professor Dr. A.K. Singh, who is now currently director of IRI, and some of our scientists from DRR in Hyderabad, they in fact developed late blight resistant lines of rice through molecular breeding. But then there are several mechanisms, several genes. So few scientists, and they have already developed actually 2 mega indica rice varieties, named this. They are resistant to leaf blight, developed through CRISPR-Cas9 genome editing. And see, it's a beautiful particular pathogen. This is Xanthomonas, which is the bacteria. It produces certain transcription activator-like effectors. This pathogen produces these effectors, which they bind to a particular promoter sequence of these genes, which are plant genes. These are sweet genes. They produce sugar. These sugar transporters help, help survival and multiplication of these Xanthomonas. So can we mutate these sweet genes that these effectors are not able to bind to sweet genes, and plant will in turn, even if this pathogen will attack, the plants will remain resistant. And that's what they did a lot of bioinformatic analysis. So many different strains of Xanthomonas they found, and ultimately they decided that there are certain sweet transporters, they enhance bacterial virulence and lead to disease susceptibility, I said before. But then scientists, they edited particularly sweet, there are 21 such genes in rice. 3 of them they mutated, and mutated where? I said before, they mutated not that non-homologous end joining type of dependent repairing, and they mutated, or even non-homologous end joining is also possible. They mutated specifically those genes, those wherein this effector, this transcription activator like effectors will bind, because when they bind to those promoter sequences of this, sweet, its transporters, sugar transporters synthesized, and they will allow these Xanthomonas to produce or multiply and cause the disease. But now when they have been mutated to bind to these sequences, and they will not make any more sugar transporters, and the plants will be becoming resistant. So this is what shows that these bacterial blight susceptible plants, 3 different varieties. This is one Japonica, and these are Indica. CRISPR editing of 3 different genes individually. They have done sweet 11, sweet 13, and sweet 14. But then multiplex CRISPR editing is also possible, I said before. And in one of those varieties, Kitake, genes. So this will be certainly highly resistant than to where only individual genes have been edited. And in fact, this is coming out to be another new fact. You know, by this time, scientists from Giri had sequenced 3,000 rice genomes. 3,000 rice genomes, and I got at least 400 of them, which happened to be the rice of Indian origin. When I was in NBPGR, I requested them to give me the sequence data, which they did. And thanks to them, 400 are of Indian origin. And those are available with Indian scientists, and I'm sure they're taking this benefit further. But then what was found, particularly when you compare the sweet genes, which are 21 in rice, there is also a natural variation occurring. And that natural variation occurring in the sense that that will also act as a mutated gene, will not allow these effectors to bind, and it will also give resistance. So there is a natural variation in the sequence of these sweet promoter genes, and resistance to a variable level, of course, to bacterial leaf blight. So, so this discovery gave us this kind of a clue that there is a natural genetic variation as well. From this, actually, I will move on to another aspect. Before that, I show you another example of loss of function of one, powdery mildew locus. Here, this is the wild type, and here's because of loss of function through genome editing, these plants become totally resistant to this fungus, powdery mildew, and this is in case of tomato. But what I was, and then regulatory point of view, I was talking to you before, that in March 2018, years ago, USDA declared that if these plants could otherwise be produced through traditional breeding, I said mutation breeding, then they will not be ready. So that's a very big advantage. And as a result, in fact, few of the crops have already been commercialized in US, particularly mushroom, I'm aware of it, and few others are also. This is a big advantage. India and many other countries are developing those guidelines for regulating genome edited crops. I was there, a member of the committee from the National Academy of Agricultural Sciences, to trying to argue that the one kind which I said before will only be mutate certain genes, for the loss of function using cell's own machinery. I hope that the Government of India, Department of Biotechnology, and others will all recommend and will agree to that. And there will be a minimal regulation as we go further into mutation of the defined sequence or adding entirely a new gene altogether. Of course, that's a separate story. So therefore, friends, what we are moving towards with recent advantages in plant biotechnology, a whole lot of technologies available, and we can try to make use of them. But then if I go back to this slide where we were talking of sequencing 3,000 rice genome, that includes national genetic variation available, that we only came to know, only from 3,000 rice genome sequences were available. Earlier, it was only Japonica, which was rice, and we thought that whole reference genome sequence of rice is enough to tell us all about what kind of genes are there, what kind of genetic variability is there, but that turned out to be not. Exists on a large, in a large way. Here we're talking of 3,000 rice genome. In India alone, we have in NBPGR gene more than 1 lakh different taxations of rice. And now, friends, the whole world is therefore moving towards Pan-Genome, which has become a new reference. Earlier, single genome sequence of a particular variety of a crop was considered as a reference. But if you, for example, in rice, there are 100,000 different excisions of rice. And if I'm able to sequence all those 100,000 excisions of rice, you will find there's a lot of genetic variability, natural proving to be useful for providing resistance to various diseases or kind of, high yield ability. Many other traits we can look for, even particularly when national resources. So here, friends, what is this Pan-Genomes all about? Pan-Genome represents the genomic diversity of a species, whole species. Two types of genes they're talking about it. Core genes, which are present, let us say, in all 100,000, accessions of rice. Those are core genes. They're all conserved. But there are other category of genes which are highly variable, which are not conserved, and they find that, yes, they are associated with certain agronomic traits for biotic and abiotic stress tolerance, So these two kinds of genes, they're called as core genes or dispensable genes. Core genes, the one rice plant as a rice plant, okay, which is conserved across as many genetic variability naturally occurring you have, it's there conserved. There are things that without that also rice plant will look like rice, okay. So these dispensable genes, they are partially shared or individuals, there could be absence or presence. They call it as a presence absence variation, In some of the species, the genes are present. In some of the species, the same genes, and genes could be related with resistance to biotic and abiotic stresses. So therefore, there's the sequencing or information of those genes, dispensable genes, become much more important. Here, there's an example of, again, shown here in soybean. See, one of the species which is susceptible to a cyst nematode, 31.2 kb, which is susceptible. But there is another variety wherein there are more than one copy and they are found to be resistant. So if you take this as a reference genome sequence, you find that these varieties are susceptible. We don't have any, kind of a genetic source of resistance occurring in soil. If you see a large number of them, you come to know that no, there are varieties or there are accessions which have resistance as well in them. So what I keep telling actually when I was in NBPGR, on a wealth of information in India, the best ever in the world. We have a second largest gene bank of the world. And I was always advocating that we must try to use sessions in different crops what we have. Breeders have their own limitations, I do agree. Certainly they're using that kind of variability, and we have to still, even in farmers varieties, land races, wild species, to the largest possible extent, molecular tools allow us. And how pangenomics became important, sequencing came down drastically. Then instead of sequencing only one rice line, now you can do 3,000 rice lines. In fact, I had a dream myself. I'm talking about 5 years. Why can't I do 10,000 rice genome sequencing? Rice genome is easier simply because diploid size is smaller. Therefore we talk of rice in the ability, and even for the nutritional point of view, there are certain genotypes of rice which become very important. And also, by the way, there are certain genes which have been cloned by outside India people from Indian varieties, Indian land races, and for example, for submergence tolerance, for phosphorus tolerance deficiency, etc., they all do in India. So therefore, this pangenomics is very important. And here, what I'm trying to show you is this, that what is that kind of the percentage of those core genes versus, if you, in case of rice, suppose you sequence only 3 accessions, the percentage of core gene is 40%. Percentage of core genes is 92%, and dispensable gene, and these are the total number of genes. But if you sequence 3,000, then this number of genes increase from 40 to 48,000. From 92 come down to 58%, and then we come to know the dispensable genes, which are variable genes, which provide all kind of resistance to those biotic and abiotic. So it depends on how many accessions we are able to. There must be some kind of optimum level, which I don't know yet myself. Not that directly I'm working into this area, but it is something to bring some of these latest advances in taking place in the world in biotechnology. And this is also true for Brassica napus, or in Solana, 53 accession sequence here, 725 sequence. These are percentage of core genes, the percentage of dispensable genes, you can see here is quite high again. And that's because we are sequencing large number, which is still much less than rice. So what I wonder that, I don't have time, but we develop a core, accessions based on morphological features. Can that core be whole sequence that will give you a total genetic variability at the DNA level as well when we talk of these dispensable genes? Well, I think I will take another 10 more minutes to request the organizers to allow me. The next, possibility of targeting gene insertion is chloroplast genome. Myself, we developed first time a report in case of eggplant, and we showed, the plants which were transgenic or resistant to a particular spectromycin, resistant, put some genes and to make them tolerant to moisture stress, in case of eggplant. But then there is a wonderful way now forward, which is not feasible through nuclear transformation, but which is easily possible through chloroplast transformation. This is a wild type, totally damaged by Colorado potato beetle. This is the nuclear transformed line, wherein one of the essential genes of the beetle itself is mutated and will not be able to grow. But nuclear transformation, that antisense doesn't work as good, but as much it does in case of chloroplast transformed double-stranded RNA. This is kind of 80 to 90%, kind of, healthy plant, or there's only few percentage damage. Here, all dead. Here it's about 50%. Here there's only 5% damage. And not only chloroplast transformation for some of these agronomic traits, even low volume, high value compounds. Currently, one of my students is working on production of few of these high value compounds by extending the pathway of carotenoid biosynthesis, and we utilize whatever the levels of lycopene or beta carotene is available to extend that pathway by the addition of few genes which are not otherwise present in the plant, and we're trying to overexpress them in chloroplast and see whether we can convert them all into some of these high value compounds, which is really very, very high value when you talk about human health, culture, etc., etc. is very, very important. And finally, when you talk of chloroplast transformation, I've got 2 more slides, that even through nanoparticle transformation is also very much possible. When they've used, chitosan based complexes of single walled carbon nanotube carriers. I mean, these are made a complex with single walled carbon nanotubes. I am not a nano biotechnologist, by the way, but this is a great piece of work done by with one of our great friend scientists, Nam Chua, who in fact was the foremost player in plant engineering when we started in the 80s. They have demonstrated that this kind of a complex can, through the cell wall, through the cell membrane, and through the double envelope of the chloroplast, and can precisely be infiltrated into the whole leaf, of a mature plant, and can produce those kind of proteins which you want to do it without stably integrating or without any physically aid or agrobacterium or ballistic gun. It can be simply on this, and this is a very good discovery, I would say. And similarly, this is the chloroplast. We knew as plant physiologists or molecular biologists that certain proteins, when they are synthesized in cytosol, they get translocated into the chloroplast with the help of a transient peptide, which is about 25 amino acid sequence. Here in chloroplast, though they are synthesized in the cytosol, they have that kind of a sequence, precursor sequence of 25 amino acids before it. So utilizing that sequence, they can deliver some kind of a agrochemical or a chemical cargo, into the chloroplast, precisely as they have shown in this paper in Nature Communications very recently. And this is the kind of a sequence they've used, the transient peptide sequence of one of the large subunit, small subunit, I believe, of and that gene, they have taken it, the small subunit, and they've shown that it has got advantages of studying more cell biology in general. Also, some of the agrochemicals can be precisely transferred, translocated actually, wherever in the plant cell or plant leaf that they require. So, and effects or will be more. So, with some of these, opportunities, we have these latest developmental. There are a lot, but in a webinar like this, examples with the limited time available to me. But then I thought this is something I should apprise all the students who are pursuing their career. I'm sure they have got a very still waiting, and fortunately they are much younger. And I wish we were also there to compete with them, but nevertheless we are here to still technologies for a country like India. And first of all, for future prospects, what I try to mention here with regard to still GM crops, we should not forget about GM crops. Producing the agrobacterium-based method or ballistic gun. And as I said before, in India, we have got about another 12 to 20 crops in the pipeline for various, ICR, CGEB, or different private sector institutions and all. We have got a large plethora of some of the very useful genes and traits, which I didn't mention to you about. The system they've all gone through, and still they're allowed. That's what I hope, for the country's benefit. Then potential of new breeding techniques, with few examples, is enormous. There are several labs in India, Banana, National Agri-Food Institute, in Mohali, in IRI, the proposal ourselves in few crops. Then the vast potential of genome engineering needs to be realized. Not very many labs in India have taken the benefit yet, but I believe they want any basic ingredients or kind of, I can assist them for sure. And then nanoparticle mediated gene or agrochemical precise transfer to plants. But this is something where we have to still wait because of the toxicity of the nanoparticles that need to be demonstrated, and not very many studies, though there are guidelines again for the regulation of nanoparticles. And then what is finally needed again, when you talk of even genome edited crops, the guidelines are, I believe, already framed, but we need that they should be totally science-based. Utilize. Why I know that social science is important, all public, all important. Our health is important, livestock important, environment is important. But the truth, science only can take you forward in life. Science can only give you the best, highest economy in the nation or globally. I've given you some of the examples of GM crops. And therefore, this is important that we utilize these new technologies to farm agriculture, which is in the interest of the farmers' welfare, consumers, and the nation. And what I talk to you is only about the tip of the iceberg. A lot one can talk about it. There's a lot one can deal with it. Therefore, I believe in a country like India, scientists, youngsters must be engaged with talking about tools like genes, genomics, genome editing, etc., because there's a long way to go, and for direction it is possible, because we are an agrarian society. More than 50% of people thrive on agriculture, and we must take it a challenge to use any such advances in technology and for human resource development and for getting good jobs outside the world, wherever. I think these technologies will ever remain important, and we should be also partnered this. I didn't talk to you about base editing. Many such tools are also available today. So with this, friends, thank you very much. Thanks to Dr. Vinod Jonkar, who always gives me an opportunity, which I like it, but unfortunately, I don't have any physical interaction this time, with the young students, and I won't be able to interact with them as much as allows us this technology. This is also another new technology, by the way, which allows us to be together, doing, presenting and getting to know about each other. So with this, I thank you all, the audience. I hope I was clear, I was audible. But if there are certain questions, of course, I request Dr. Rakesh to kindly help me. Sorry, in the beginning, there were some hiccups, but I think you managed, but somehow this logo keep coming all the time. Never mind. I can understand the other technological difficulty there. Very much for this opportunity. Thank you.
Thank you very much, Professor Bansal, for the brainstorming session.
These
Sure.
are genetically modified. So thanks with genetically modified flowers.
Yeah, thank you, sir. Thank you. I'm sure audience have really liked the presentation, and it was a really beautiful and knowledgeable presentation from your side. And it looks like we have few questions in the chat box. I'm reading them up for you, sir. Am I audible to you, sir? Hello? Professor Bansal, am I audible to you?
I was in a webinar.
Hello? Do you get my voice?
Yes, please. Yes, please.
Yes. I'm reading question for you, sir.
Hello.
One audience is asking, with genetically modified organism, there is introduction of foreign genetic material, hence referred to as transgenic, whereas with CRISPR-Cas involving altering original base pair arrangements within genome of an organism, can transgenic term be used in this case?
See, the trans is from any other organism. So when you are not bringing any gene from any other organism other than your own gene, we will not be calling it a transgenic plant. So this is the difference one try to learn. Trans is the word from where it is outside that particular world. For example, America is a trans country for India, So if I take any gene from rice and put it in wheat, the wheat gene is coming from another source altogether, or bacteria or any other living organism. But here I am mutating precisely plant's own genes. If I'm editing in the sense, I'm putting few bases or deleting few bases out of the whole genome, or a gene of a plant, I'm not putting any other extra or genes into it. So we call it as not transgenic, genome edited, plants.
Next is Dr. Deep Shikha is asking, are there examples where GM has not delivered the promised improvement in crops? GM crops have only been around for 20 years. Might there still be unexpected and untoward side effects? What can be done to prevent crossbreeding of GM crops?
See, phases of transgenic crop development. Okay, we produce hundreds or even thousands of times events, okay, with a transgene of, we select only 5 to 10 events which are best performing and which are phenotypically, morphologically same as the and they are tested for the expression of the trait in small field trials and large scale field trials over a few years, and based on all that analysis is selected and then commercially released. So all these kinds of testing are always done. So one should know, there is a whole procedure. We have to go through it systematically, this is what is biosafety regulatory guidelines. You cannot simply release anything which has or which is not showing that particular agronomic trait of expression to that level, even if it is BT resistance and the level is only 50%. No, that cannot be allowed. It has to be of that particular insect before that is allowed to be released into the commercial scale to the environment. So these are all taken care, actually speaking. There have never been, even among those crops which have been released commercially, anything of that kind has been reported scientifically, not yet.
Yes. Next question is, sir, after the start of consumption of GM crops, up to what maximum age the effect of human has been monitored?
Me, I believe, according to me, you know, there are tests, already well-defined tests which are done, and there are no tests done directly on products. Or even, for example, you know, for some of these products, you know, pharma products, they're done on rodents, they're mice, they're you know, rabbits. They are all done based on the kind of a cycle we have of human beings, you know, and there is no such test of GM crops required to be done on a long-term basis. Only tests which have been done, you can talk about it, of course, 20 years test if you take it from 1996 onwards, now 25 years completing almost. If we have 450 million population of US alone is consuming, we are also consuming in the form of, let's say, BT cotton seed oil, which is a part of vegetable ghee. In countries, those who travel outside, means millions and millions of people are consuming, and we have not come across any such effect. So that's kind of a long-term effect. One can talk about it, you know, there's no such adverse effect or side effects been observed yet.
Next is, even though numerous applications of GMO, there is a fear that farmers might rely on GMO company for GMO seed procurement year after year. How could we resolve this fear?
I think, you know what, if I talk very plainly, we seem to be more worried than farmer himself in this regard. Year after year, farmer is very much willing to buy seed, new seed year after year, provided it gives you that advantage. In case of BT cotton, are buying seed year after year for the last 18 years. Right from 2002 until 2020, the area almost 95% under BT cotton. Whether to get that benefit which is promised to them. Even if the hybrid which is non-GM, any hybrid which is non-GM as well through hybrid seeds have to be bought anyway year after year by the farmers, and because it gives you advantage of 25 to 30% more yield, you know, with even little extra input, balance sheet is that he gets more money, more remuneration, and therefore he would prefer it more. So that kind of a fixation of, you know, buying seed every year and after year, farmers want only thing good quality seed, the promise, you know, which we give to them, that must be there, you know, with in fact timely input of fertilizers, timely input of insecticide, whatever is required. Farmers are as entrepreneurship as anybody will think about it. So they would like to invest, they would like to harvest more benefits and money out of it, and the welfare for them and for their children.
Dr. Sushmita,
so this is not that we need to worry about year after year purchasing the seeds.
Yes. The next question is from Dr. Sushmita Shukla. She's writing, can you please let me know any of the genes showing resistance against biotic stress which may work for raising GM Vigna variety?
Which disease?
Vigna.
I couldn't hear.
Sir, I'm repeating the question. Can you please let me know any of the genes showing resistance against biotic stress which may work for raising GM Vigna variety?
Yes.
See, first of all, she has to define what biotic stress, which type of biotic stress.
It is not
Vigna can be affected by several diseases, whether it's a fungal, bacterial, and whether that molecular mechanism is studied well to find out the, you know, the candidate genes. There are very many approaches. I would suggest if she can write to me, my email was given, or if you can pass my, those who are willing still to continue dialogue and ask questions with me, you know, sure, they can do it through email. My email is, otherwise, if I may just right now, name kcbansal27@gmail.com. Happy to see you, Dr. Kothari.
It was a wonderful talk, if I can speak for a minute, and very informative, and everybody, every biologist need to know all these things. And what genome editing, gene editing, benefits can it add to the agriculture and other areas.
Yes.
Yeah, also Professor K.C. Bansal, and I know him personally for last many years. My question, Professor Kailash Bansalji, is
Yes.
which groups in India are CRISPR technology?
Actually, Dr. Kothari, DBT has taken a lead in this now to, give some project. At the moment, very few. See, if you talk of rice, if you talk of crop wise, I can tell you,
Yeah,
Dr. Vishwanathan at ICAR,
yeah.
he has already published one paper. I refer to the DST gene in rice. Then banana, I think from National Agricultural Food Institute, NABI, and then I think ICGEB, probably they're call, or Dr. M.K. Reddy, and we are now actually writing in a particular crop. I'm not revealing that. 31st August.
You know why I asked this question?
Yes.
Because this is the technology for the future.
Yes.
The way India developed GM technology in this country, we put to good use. But
Right.
technology, CRISPR technology, should be developed with the same vigor with which we developed GM technology.
It's far better than GM technology in terms of regulations, precise accuracy, time factor, and not bringing genes as there was hue and cry. You bring genes from anywhere. No plant has its own genetic. Capture that, know it at the scientific level through genome sequencing projects, and then there's a lot. I tell you, we're talking about today. India's food production 295 million tons.
Yes, yes.
I tell you, we can easily go to 350 million tons by next 10 years.
Yeah.
Mission mode approach, concerted effort by Government of India, and giving that kind of regulation eased out. Precisely for the benefit of the nation, one can do it.
Yeah.
And human resource development to a larger extent, youngsters.
People ask question on behalf of farmers, which farmers do not raise.
Exactly.
Are interested in GM.
You're right.
And
I would expect. And that's what I said. If they don't ask questions now, they won't be able to learn. And I give them a freedom of writing to me through emails. To my stability, I will try to ask the questions. The farmer. Let the science drive the way forward.
Sir, good evening, sir. This is Dr. Shushmita Shukla. I was just wanted to know about the Wi-Fi. Writing a separate email for that, sir.
Sure, please write an email. Please write an email, Dr. Shukla.
Thank you, sir.
Most welcome.
Thank you, sir.
Next question is from Anjali. She is asking why cotton absorbance property decreased when BT gene introduced.
Pardon me, say it again.
The question is why cotton absorbance property decreased when BT gene introduced.
I have no idea. I have not question before or any such observation by any other person. I have to do it or study myself, and I don't know whether that is the case. It has been investigated by the scientists themselves, but I'm sorry, I have no answer for it.
Scientifically speaking, this kind of possibility is.
To me, it also appears. Otherwise, we would have known it by now.
Next question is I think this is the last question of the session. Pramod Kumar is asking this question. During the gene editing or mutation, what type of metabolic perturbation you have experienced in your experiments?
Dr. Hari mentioned, we have not done any genome editing experiments ourselves yet, but in the literature, there's no metabolic perturbations affected. Obviously, engineering to do it. if you are trying to, let us say, change a particular metabolic pathway, precisely on that particular metabolic pathway connections, and some perturbations are certainly expected. But I don't think that people have gone that far. I have no example at the moment before me with that I can talk to you about this. And to me, it appears the possibility would be least. Nevertheless, there could be also a minor possibility if the gene product or a metabolite happens to be a particular metabolic pathway. The kinetic activity of a particular enzyme. We don't know that unless some metabolomic studies or otherwise are downstream carried out. So far, what we have come across through genome editing are some of these principal metabolomic traits, which have been incorporated precisely. In case of tomato, beautiful example of bacterial blight. In case of in case of rice. So I'm sure there are certain downstream mechanisms of action need to be studied. Able to answer this question.
Thank you, Professor Bansal. I think.
Thank you very much, sir. Thank you very much.
Now I would like to request Dr. Sandeep Kumar to offer vote of thanks to the speaker and the audience. Over to you, sir.
Thank you, Dr. Rakesh Yadav. Thank you, Professor Bansal, for your wonderful lectures. So it is always a pleasing experience to listen to you, and I have got the opportunity to interact with you a couple of times and listen to you. So it is always a very pleasing experience. So I'm sure the participant must have been benefited by your rich expertise in this area. On behalf of the department, on my personal behalf, and especially Department of Biotechnology, Government of India, so I'd like to place on records so our sincere thanks to Professor K.C. Bansal for sparing his valuable time and educating our students. So I'm sure they must have been become more knowledgeable through this online webinar. So actually, we do miss some kind of physical interaction with you, sir. So I'm sure in the coming time, so when these restrictions will be over, so we'll try to have you in our department. You are associated with our department, for a significant time now, and we miss your visits. In today's lecture, so I really appreciate the way you highlighted some of the recent trends, especially related to gene delivery using chitosan complex single-wall carbon nanotubes, and some of the routes of delivery of agrochemical using nanomaterials, and then citing some of the important aspects related to nanotoxicity. So I am sure that the entire deliberation today must have increased the knowledge of all the participants, and I really thoroughly enjoyed your lecture, and I look forward to hear you again, time and again. So thank you. So I like to thank all the participants who are present here, so in this webinar series. And once again, I thank Professor K.C. Bansal from the bottom of my heart to spare his valuable time. So again, thank you one and all. And now we'll close this. Thank you, Bansal sir, again. Okay.
If there is any feedback, I would like to know about it, please. Thank you, Dr. Kothari. See you later.
Thank you.
Yes, sir.
Thank you very much.
Thanks to all my friends at GAU, Dr. Vinod Chonkar, and everyone else. Thanks to all of them.
Thank you, sir.